and dear students, in my previous lecture, I have already started regulation of gene expression in the yeast. Regulation of gene expression in yeast, and this is a very important topic which is included in paper third of biotechnology part 2, molecular biology syllabus. So this regulation of gene expression is very important because the mechanism of regulation of gene expression in the yeast is brought about by the process of transposition. Transposition. What is transposition? I have also described it. Transposition is defined as the change of site or change of position of one gene from one site to another site on the same chromosome, or from one chromosome to the another chromosome. As you know that genes have their specific site located on the chromosome, and that sites are known as the loci. Okay, locus. And okay, so the genes are present at the locus, but there are some genes which can change their position on the chromosome, and such genes are known as the jumping genes or transposomes. Okay, and the phenomenon of change of position from one site to another site is known as the transposition. So this transposition is very important, and the regulation of gene expression in the yeast is controlled by this mechanism, this transposition method. So as you know that the you have already studied yeast, and you know about the yeast. Yeast is a unicellular Ascomycetian fungus, and in my previous lecture I have described that yeast is a haploid, and the haploid yeast are of two types: alpha yeast and A yeast. There are two different forms of the yeast, alpha yeast, which are known as the alpha yeast and A yeast, and these two yeast are interchangeable. That is, one form of the haploid yeast can change into another form of the haploid yeast, and vice versa. And the yeast can also grow in the diploid condition. You know that the yeast can happily grow in both the haploid and diploid condition. So you are not required to write the conversion of the haploid yeast into diploid yeast and diploid yeast into the haploid yeast. The purpose of the study of the regulation of gene expression is to study the mechanism of change of one form of the haploid yeast into the another form of the haploid yeast, which is known as the alpha yeast and A yeast. Okay, so this A alpha yeast and A yeast, these two yeast, alpha and A yeast, and it is very important to note here that either it is alpha yeast or A yeast, both the yeast have the genes for making alpha and A yeast. Yani if both the form of the yeast, whether it is alpha yeast or A yeast, both the yeast have the genes on their chromosome for making alpha and A yeast, but the genes present at their original site are not active. Rather, when the genes are transferred to the expression site, the gene expresses itself, and it leads to the formation of A yeast or alpha yeast. So on the chromosome of the yeast, there are three sites: alpha site, A site, and mat locus. Mat locus is very important, which is present in the central position of the chromosome, and as shown in the diagram. And what I am teaching it will become very clear if you go through the diagram which I have provided in the form of the PDF prior to the commencement of this lecture. So at first you must go through the 4 pages in the form of PDF file which I have loaded in this WhatsApp group, and that will help you to understand this. So just I am talking about the presence of the three sites on the chromosome of the yeast, and these three sites are the alpha site, A site, HML alpha site, HML A site, and the mat locus. So the gene, and just I have said that both the alpha and A yeast have the genes for making A yeast and alpha yeast, but the nature of the yeast, whether it will be alpha or whether it will be A yeast, will depend upon the presence of the gene at the mat locus. Mat locus is called the expression locus, whereas the alpha locus and A locus are known as the silent locus, because the genes are not expressing there at the alpha locus and A locus, and therefore they are called the silent locus. And when the gene when the gene moves, or when the gene moves to the mat locus, the expression of that form of the gene will take place. So this is very important to note that if there are two sides on the chromosome of the alpha and A, which are known as the expression site and silent site. So silent sites and silent site and expression site. The expression site is known as the mat site. So if the at the mat site alpha gene is present, what will be the nature? If at the mat locus alpha gene is present, the yeast will be alpha yeast, and if the A gene is present at the mat locus, the yeast will be A yeast, and one form of yeast can change into the another form in this way, that when alpha is, if suppose that if the alpha is to change into the A yeast, what will happen? At first, the alpha gene from silent locus will be copied, and it will be transferred to the mat locus, and it will get attached at the mat locus by replacing the genes present earlier there, and that will now and at the mat locus the gene will express itself, alpha gene will express itself, and it will convert the A yeast into the alpha yeast. So this is the method by which the A yeast gets converted into alpha yeast. Similarly, if now the alpha yeast, again if the alpha yeast has to change into the A yeast, what will happen? The gene A, which is present at the silent locus, is copied, and a copy of it is transferred to the mat locus, and at the mat locus it replaces the earlier alpha gene, and it gets attached at the mat locus, and it expresses itself, and its expression converts the A yeast into the alpha yeast. And the very important point here is that the alpha yeast cannot change into alpha yeast, and A yeast cannot change into the A. That is the transposition of alpha into alpha and A into A do not take place. Rather, alpha yeast changes into A yeast, and A yeast in turn changes into alpha yeast. So this is the method. This is the method by which the alpha yeast changes into the A yeast, and A yeast changes into alpha yeast. And this process and this mechanism and this mechanism of conversion of the mating type, there are two mating types. You must know that there are two mating types in the yeast: alpha mating type and A mating type. You will find in the note that the word mat, MAT stands for the mating locus. So there are two forms of the alpha haploid yeast which are known as the A alpha mating type A and mating type alpha, and these two mating types are interchangeable, interconvertible, and this interconversion is catalyzed by the genetic process that is known as the transposition. So in this way, all the 4 pages which I have uploaded in the notes, you must go through and try to understand. And if there is any problem, I will discuss all these things in the next lecture, and I will also provide a note. Just I have provided you the 4 pages which shows the diagrams, diagrams of the yeast chromosome, where the silent locus and mat locus are present. And the first page of the notes provided today include the conversion of haploid yeast into the diploid yeast, and diploid yeast into the haploid yeast. You have nothing to do with that conversion of haploid into diploid, and diploid into haploid into diploid, and diploid into haploid. Your purpose is to study the conversion of the alpha yeast into A yeast and A yeast into alpha yeast, which is, and this the genetic sketch of this conversion of A yeast into alpha yeast is present on the page number 2, where you will find the A locus and silent locus and mat locus on the right side of the chromosome, and the left side of chromosome you will find the silent locus, whereas in the central position you will find the mat locus. and the nature of the yeast will depend upon the presence of the gene on the mat locus. If the alpha gene is present at the mat locus, the yeast will be alpha yeast. If the A gene is present at the mat locus, the yeast will be A. Why it is so? Because at the silent locus, HML alpha locus and HML A locus, the genes are not expressed themselves. Genes are not capable of expressing themselves. They will remain silent there. When the gene will change its position, when the A will change its position from HML A side to the mat side, the gene at it is only the mat locus. Remember it, the very important point. It is only the mat locus where the gene expresses itself. So this is the very important, and this question is generally asked in